Now that we've refreshed your memory on the structure of DNA, we can now begin to discuss how DNA actually stores information. I mean, think about it. These molecules inside your cells are supposed to hold enough information to tell each cell what kind of cell to be, to control its activities, and to control the activities within your body over the entire lifespan of your life. Unless you have an identical twin, your DNA also is unique. It makes you genetically unique among all other life forms. No other form of life has your exact sequence of DNA. So where is this remarkable information stored? Well, let's look at a section of DNA. A section of DNA such as this might be called a gene. And a gene is really a recipe for making either an RNA molecule and or a protein. So we can make RNAs or proteins based on this information. Where's the actual information? Well, it's here. It's in the bases. Remember, it's not going to be in the backbones because the backbones just repeat over and over. But the bases have a sequence, and it's the sequence of those bases that's important. What the cell will do is it will open up DNA, it will choose just one of the strands, and different genes have different strands with information, and it will read this strand, for instance, from top to bottom. It'll read the sequence adenine, cytosine, guanine, thymine, and so on. And because the sequence of those nucleotides differs from one gene to another, that can store information. Think about reading a recipe in a book. The order of the words used in that recipe is really important. If you change the order of the words around, it can change the meaning of the recipe. Same thing applies here. It's the sequence of the bases that stores the information. At this point, if you're thinking that it's pretty implausible that a molecule as simple as DNA could store all of that information, you're definitely not alone. Back in the 1950s, when DNA was discovered, scientists were shocked at how simple it was. I mean, it's only made of 4 different types of nucleotides in these repeating patterns. So scientists thought, well, there's no way that DNA is complicated enough to store genetic information. So for a long time, they thought that something more complex, things like proteins, which are really complicated, must be where our heredity lay. And yet, it turns out that DNA really does store that amount of information. To explain how this works, I want you to think of DNA as being a molecular language, a chemical language, and it's a language that only has 4 letters. Now, 4 letters isn't very many letters, especially compared to a language like English. English has 26 letters, and think about all the things we can express in words using our combinations of 26 letters. Well, English is a pretty complicated letter or language, and there are simpler languages. Consider this: Hawaiian only has 12 letters in its alphabet, and you can express anything in Hawaiian that you can express in English. There are even simpler languages. Binary language, the language of computers, really only uses 2 symbols, zeros and ones. So there's an even simpler language that only uses 2 characters to express things, and you can express everything in binary that you can in English. So a language such as DNA actually falls kind of in the middle. With only 4 letters in the alphabet, you can express everything you can express in English, essentially. And you can express all of the information you need to run a cell and to run an organism. Incidentally, I still occasionally hear this argument that DNA is too simple to store the genetic information of life being tossed around. Not among scientists, mind you, but by groups that, let's say, just wish evolutionary theory could be disproved. The idea is that DNA is too simple to store all of the genetic information that makes everything from giraffes to fungus to bacteria to people, so that some sort of a creator or an intelligent designer must have been involved in the formation of life on this planet. Now that you know a little bit more about DNA, I hope you can see the logical flaws in this argument. Even though the language of DNA only consists of 4 letters, you can still store a lot of genetic information using those 4 letters. The secret to this system is really in the sequence of those letters, not in the number of the letters. Let me show you an example. I'm going to draw out a sequence of nucleotides that is 10 nucleotides long. These are A's. They may not all look like it. There we go, 10 A's in a row, 10 adenines in a row. And let's say that I was just going to try and see how many of these sequences I could come up with that are 10 letters long in length and are only use those 4 nucleotide letters. Well, here, this first one is all adenines. Maybe the second one is 9 adenines and a thymine. Maybe the third one I come up with is 8 adenines, a guanine, and a thymine, and so on. If I kept doing that and was really, really persistent, I could come up with over 1,000,000 sequences that fit those rules, that are only 10 nucleotides in length. Now consider this. The sequences that code for most proteins and RNAs aren't 10 nucleotides in length. They're hundreds of nucleotides in length, or even thousands of nucleotides in length. When you have a sequence of that kind of size, and you're looking at the sequence of the letters involved, you can code for a whole lot of different kinds of proteins and a whole lot of different kinds of RNAs. You can literally code for millions of different things because the sequences differ. We're going to look at how the cell actually extracts information from these sequences more in Chapter 17, but I'll give you a little introduction for now. In our chemical language, I want you to think of the individual letters as the nucleotides. So the A's, T's, C's, and G's, or A's, U's, C's, and G's, those are our letters of our language. If you put 3 of these letters together, you get a word in this language, something called a codon. So what the cell will do is it will read these sequences, and it reads them in groups of 3. And each group of 3 means something. Each group of 3 codes for a specific amino acid. And if you put the right amino acids together in the right order, you can make a specific kind of protein. This entire sequence with all of its letters and codons might be one story or one recipe in a book. So that little sequence is called a gene. Again, a gene is a recipe for making a specific protein or sometimes RNA. One thing that I hope you take away from this lecture is really just how simple the structure of DNA is. It's really just 2 strands of nucleotides that fit together in a very predictable pattern. However, even though the structure of DNA seems simple by today's standards, the discovery of DNA, that story, is far from simple. The discovery of the structure of DNA was really the Human Genome Project of the 1940s and 1950s. And by that I mean that there were a number of labs and many scientists trying to figure it out, and they were all competing with one another. Now, each different lab was able to figure out a little piece of the puzzle, but no one could really put together the whole story and figure out the whole structure until these 2 guys named Watson and Crick came along in 1953, and they put together all the information and figured out the overall structure of DNA. Now, this story is very controversial because Watson and Crick essentially stole a lot of data from other scientists. Some of it was shared with them legitimately, and some of it they just stole. They were known for doing things like slipping into the back of research presentations and making a few notes and slipping out before anyone knew they were there. I really don't do this story justice, so what I'm going to have you do is do some reading in your textbook in Concept 16.1, and I'm also going to have you visit a website called Vision Learning and read about these different scientists, read about their different contributions, and take some notes on that. So that is included in your guided notes, and I do want you to complete it before you come to class next time. We're going to work with it some more. All right, you guys, you're finished with Chapter 16, Part 1. Make sure you complete your notes, try all your turns yourself before you come to class, and then bring that stuff to class so we can dig into it and go over it. Good job.